Good morning, students. Gene therapy. It is the process in the bond with the hereditary disease and the correcting therapy being given for each disease. Yes, this is the gene therapy. It's very important question for your public exam. So please, you have to note down clearly therapy, that is treatment. We already know that. So it is the process. What is the process? Gene treatment, that is we call as a gene therapy. Okay, expression of the normal gene, the person's result in the function and gene products whose active products of the normal phenotype, and transfer the normal gene into person cells that carry one's mutant alleles. Okay, next gene augmentation therapy, that is a treatment. How it is that it will be a transfer normal gene into the person cells of the carry from that, and the gene inhibition will be happen in that. Gene augmentation therapy treatment also involved in the DNA into the genome to replace the missing gene product. The two approaches are a gene therapy is that, and the somatic cell therapy and germline gene therapy. Okay, so this is very important. Next is stem cell therapy. So two main types of stem cell therapy. It is the adult stem cells and embryonic stem cells. What is the adult stem cells? Adult stem cells means found in the various tissue of children as well as adult, and that it is somatic cells divided the creating another cell similar to the most of the adult stem cells are multiple and cannot the replace system of the body then replicating adult tissues. Okay, the embryonic stem cells means it is based on the ectodome, mesodome, and the cell itself it will be happen in that, and that when stimulated the ears can develop in the more than 200 cells types of the adult body. So it is isolated from the epiplast tissue of the inner cell mass of the blastocyst. Ear cells are eliminated in that they can proliferate in the sterile culture medium and maintain their undifferentiated state. Okay, so banks are there, stem cells banks are there. That banks where they are extraction, processing of the storage of the stem cell, so that may be used for the treatment in the future when required. Amniotic cells bank of the facility that stores stem cells derived from the amniotic fluid for the future use. Stem cells are stored in the banks especially for use by the individual from whom such a cell have been collected, and the banking costs are paid. Cord blood banking in the extraction of the stem cells from the umbilical cord during the childbirth. Okay, so while the umbilical cord and blood are the most popular source of the stem cells, the placenta, amniotic sacs, and amniotic fluid are also rich source of the terms of the both quantity and quality. So this is a molecular diagnosis. Each molecular infecting disease are inherited disease and a defect in the essential for appropriate treatment. Early detection of the disease is not possible using the conventional diagnostic method like a microscopic examination, serum analysis, and urine analysis. This is only for done in the laboratory. Scientific work continuously searching from the sensitive and diagnostic techniques of this diagnostics of disease. Okay, and next recombinant DNA technology and polymerase chain reaction. These are the technological development of the enzyme linked immunological assay. There is a ELISA test. It is very helpful for the techniques for using this. We can identify this the molecular diagnostic of the disease. Okay, this is separate courses are available, BSc, MSc like that, and engineering technology also is there available. It is present in Bangalore. So if you are interested, you can go and study from the future further studies from the after 12th completed. Okay, presence of pathogenic like virus, bacteria detected only when the pathogen product symptoms in the patient. But the time of the symptom, so concentration of pathogen become very high in the body. However, the low concentration of the bacteria and the high concentration of the bacteria in the umbilical of the nucleic acid. Okay, ELISA, you know that enzyme linked the immuno acid, that is the AIDS test. So how they will do that? See, this is the ELISA test. This is the microbiologist. They will do that like that. When ELISA is a biochemical method, so there is only biochemical. So biochemical engineering is there. Though that also they are doing it in that technology. So BSc biochemistry also they did for the ELISA test also. This is how they are doing. Means this is EVA, evangeline and Peter permeation. So that he only discovered in the year of the 1971 to detect the presence of the specific antibiotic or antigen of sample of the serum, urine, etc. Serum means blood. Okay, very important diagnostic tool in the determine the person of the HIV positive or negative. ELISA is a very good tools. Okay, so they determine the serum antibody concentration. So antibody produced in the person infected by the pathogen such as HIV. So already we are discussing the practical for how we can our blood test antigen antibody. What is antigen antibody already we are discussed. So you can just recall that is only which is helpful for the ELISA test. Okay, so gonadotropin such as human chronic gonadotropin. During the diagnosis is sample suspended contain the antigen impulse on the surface of the ELISA plate, and it is antibody specific antigen allowed to react with the antigen linked to the appropriate enzyme like peroxidase. Unreacted antibody is washed away from the substrate. Activity of the enzyme is the color product indicating presence of the antigen. The intensity of the color is directly proportional to the amount of the antigen. ELISA is highly sensitive and it detects the antigen. So direct ELISA, indirect ELISA, sandwich ELISA and competitive ELISA. These are the 4 kinds of ELISA. Next class we can discuss about that. See the direct ELISA and indirect ELISA, sandwich and competitive ELISA. This diagram you have to draw in your note and you can use it. It is highly and specific for use for the diagnosis. So it's not in your book. So you can please draw in your note and you have to use it in your exam. So you will be getting more mark from this question. Okay. ELISA possesses the additive advantage of the not recording a diacritic. So it is a counting apparatus. Thank you students. Next class we discuss.